In this lecture, we'll talk about animal cloning. Animal cloning is a creation of an organism that is an exact copy of another one. So this means that every single bit of DNA is the same between the two. So clone actually is a group of genetically identical cells. A clone is also an organism which is genetically an exact replica of another organism. Cloning is to duplicate a cell or an organism, usually sexually, which is genetically an exact replica of the other cells or organism. So we'll talk about how animal cloning is performed and what are its benefits. So just a brief history of animal cloning. In 1962, John Gurdon claims to have cloned frogs from adult cells. 1963, JBS Harden coined the term clone. 1970, Carl makes claims to have cloned 3 mice. 1983, Salter and McGrath fuse a mouse embryo cell with an egg without a nucleus but failed to clone these using their techniques. 1984, Steen clones sheep from embryo cells. 1986, Steen clones cattle from differentiated cells. 1986, first Prather and Eystone clone a cow from embryo cells. So these are some of the earlier claims. But the major breakthrough came in 1996, Dolly. Dolly, the first animal clone from adult cells, it was born. Dolly was born. Soon after the creation of Dolly, so this was a major breakthrough in animal cloning. 1997, President Bill Clinton proposed a 5-year moratorium on the cloning. 1997, Richard Seed announces his plans to clone a human being. 1997, Wilhelm Cameron creates Polly, a cloned sheep with an inserted human gene. 1998, Wakayama creates 3 generations of genetically identical clone mice. So these are some brief history of cloning. Major breakthrough came in 1996 when the dolly was created. So why do we want to do cloning? Researchers hope that these techniques can be used in researching and treating human diseases and genetically altering animals for the production of human transplant organs. So this is the process of animal cloning. Step 1, take any cell from the body, from the skin for example. Take an egg cell from the ovary of any woman. Take the nucleus out of the egg cells. So from the egg cell, nucleus has to be removed. Now fuse the cell of the skin and the egg without nucleus. It will start to multiply, forming a microscopic ball of many identical cells. So in the next step, in about 6 days, place it in the uterus of the woman. In 9 months, a baby will be born just like you, an identical twin of you, without any genetic characteristics of the woman who gave the ovule and provided the uterus and gave birth to your twins. So this is how the animal cloning is being done. So here is the unfertilized egg obtained from the female, and from this nucleus is removed. And the donor cell is basically an adult cell, particularly mainly from the skin of any animal, skin cell which has a nucleus now. So this is a step 1. Step 1, the nucleus of the egg cell is removed. So then we can take the nucleus from here and put it here, or one can do the fusion. Now this is a cell containing donor cell nucleus. It takes about 1 hour for an egg to accept the new nucleus. Then the egg is allowed to sit for additional 5 to 6 hours. During this time it undergoes no development. After for about 6 hours, the cell is placed in a culture that jumpstarts cell development. So it undergoes cell division. The cell culture function in the same way as in the electric shock, but it's less strength on the cell. The egg cell develop into an embryo. Here, it's all under the egg cell and with the donor nucleus undergo cell division. It forms blastocyst, which is then implanted into the uterus of the surrogate mother. After a normal gestation period, a clone is born. So after that. So this is how the simple process of animal cloning. So this is an example here. So this is sheep, actually organism to, this is the organism to be cloned. So the skin cells and any differentiated cells can be taken. Generally skin cell as fibroblastic skin cells are taken. And similarly then alongside one unfertilized egg cell is taken, and from that nucleus is removed, and now we got a egg cell without nucleus. No egg cell with no nucleus taken. Egg cell and the cell to be cloned are fused together. Okay, so these two cells are fused together here now, and then they start to develop into an embryo. They this the, for this for egg, this after this fusion they start undergo cell division, and they develop, form an embryo, and then implanted into the foster surrogate mother. So the so the so the newborn will be same as the organism which has donated the differentiated skin cells. So it will be it will be basically the clone of this particular sheep, this particular animal. So this is the basic principle of cloning. So here is the same thing here. Here is the adults. So so these adults possible donors, one can, besides taking from adults, one can take from fetus, early embryos or blasts, any of these cells. Then one can culture it. Okay, so from all these can be donors, and then egg cells are taken from female, and the nucleus is removed. Then again the same thing, fusion occurs here. Both cells are fused together, and then you have a blastocyst. It undergoes cell division, and then it undergoes cell division and form blastocyst, and then it's implanted into, and you get a sheep, and you get the progeny. We get the offspring's progeny, which is similar to the donor. So this is the same procedure. So the advantage of animal cloning is that one can use it for curing any particular disease or some genetic disorder. So this is called purposeful cloning, means animal cloning with therapeutic purpose. Like this, one can take cells with defective genes, and then viral vector carrying the healthy genes can be then introduced into this particular cell with defective genes, and then this vector will then viral vector will then introduce the healthy genes into the cells, and the new gene in the cell along with old genes. So you have a now a healthy gene in the cell. So now this particular cell with the healthy gene or the normal gene can be then fused with the egg cell without nucleus, and then one can do the cloning. So basically to get a healthy copy of the animal. So this is useful in particular in cattle and the animals. So this is purposeful cloning, which means for therapeutic purpose. In it can be this animal cloning can be used. So application of animal cloning, animals as drug producer, gene farming. One can use animals for producing therapeutic protein in large amounts, particularly in the milk, in their milk. Animal models, model for human diseases. One can use these animals for studying human diseases. Breeding and endogenic body tissues, transplantation, particularly the pigs can be used for organ xenotransplantation. So these organs then can be used for transplantation in human. So these particularly it's done with the pig. From the pigs, organs can be taken and transplanted in human. So livestock breeding and agriculture, it can also play an important role there. So briefly we'll go through this the creation of Dolly. So this was the major breakthrough in animal cloning. So Dolly was a female sheep cloned successfully from an adult Finnish Dorset sheep memories gland cell and the enucleated egg of a Scottish sheep. So here the donor cells, the differentiated cells were taken from mammary gland cells. Born on July 5th, 1996 at the Roslin Institute at the University of Edinburgh in Scotland, Dolly's birth was the result of 277 attempted cloning by the research team at the Roslin Institute, which included embryologist Ian Wilmot and biologist Keith Campbell. So they tried 277 times. So animal cloning success rate is very low. So one has to try several times. So they tried 277 times. Only then they were able to get create the dolly. Scientifically, cloning dolly was a breakthrough, but the event had many social implications as well, casting the concept of cloning into spotlight and causing quite a debate worldwide as to the ethics of the whole process. Dolly the sheep was born on 5th July 1996. He is a scientist who created this dolly. 98 scientists in Scotland have announced the birth of the world's first successfully cloned mammals. So this was considered the first successfully cloned mammals, Dolly the sheep. It is a female domestic sheep, first mammal to be cloned from adult somatic cells using the process of nuclear transfer. They took a cell from her body and then developed it artificially so that it became a new sheep that was an exact copy of her. So this is how Dolly the sheep was created in 1996. This uses the technique of somatic cell nuclear transfer, where the cell nucleus from an adult cell is transferred into an unfertilized oocyte that has had its nucleus removed. So this already we talked about on earlier slides. The hybrid cell is then stimulated to divide by an electric shock. So they use an electric shock to divide it, and then blastocyst that is eventually produced is implanted in a surrogate mother. So here this is example. So this is how they did actually here. So here is the Dorset sheep which provide this from which the memory cells. These are the differentiated cells. Memory cells were taken. Their adult memory cells were taken. So it provided the nucleus, and the Scottish black face, which has a black face here, you can. It was a cytoplasmic donor. So in which basically the X cells were taken and nucleus was removed, and then both of them they were fused together, and then electric shock was given and the cells started dividing. It formed a blastocyst, and then it was put again back in a surrogate mother, and we got the dolly the sheep, which was quite different from the surrogate mother, and also it was quite different from the mother which provided the eggs here. Okay, so it was quite different from the but it was similar to the Finnish Dorset, from which provided the adult memory cells. So it was the exact copy of this particular sheep, the Dorset sheep here, which provided the memory cells. So this is how the animal cloning. So this was the exact replica, genetically exact replica of the this particular sheep. Basically, we can say it is a clone of this particular sheep, exact clone of this particular sheep. So this is how the dolly was created. So this is the same process here. When Dolly came out genetically identical to the white-faced sheep from which the mammary gland cell was taken, and not the black face if the egg was donated by, or the black face if that carried Dolly to term, scientists knew that they had successfully cloned a sheep using the process of nuclear transfer. So this process was called somatic nuclear transfer. So this is how the Dolly sheep was created, and it led to the development of several different animal clones. So animal cloning, after this success, several different animals were then cloned successfully. So this is just to show that what are the chances of getting a dolly, or for the chances of for animal cloning, any animal cloning. So it's cells taken from a 6-year-old finished Dorset Eve and cultured in a lab. 277 cells then fused with 277 unfertilized eggs, each with the nucleus removed. So they did 277 experiment, we can say. 277 memory cells fused with 277 unfertilized eggs. Enucleated unfertilized eggs. So from 277 fusion experiments, now 29 viable reconstructed eggs survived and were implanted in surrogate black face Eve. So only out 277, 29 viable eggs were then obtained, and from this and which were then implanted into surrogate Eves. From 29, only one gave birth to Dolly. Only out of two, you can say out of 277, only one gave birth to the Dolly. So success rate was very low. So 0.361% chance at the onset, 3.4 onset once implanted. In nature, between 33 to 50% of fertilized eggs develop. So you can say the probability of animal cloning is very low. Just a 3.4% almost. But in the nature it is 33 to 50%. So you can imagine the very low success rate of animal cloning. However, the sad part of Dolly the sheep was she died on February 14, 2003, nearly 6 years after her birth, due to progressive lung disease caused because her cells were already old, and she also had premature arthritis. She died after 6 years after her birth, because actually the cells, she was already aging, because the cells were the old cells were, nucleus was taken from the already a fully grown sheep. So she was basically not that healthy, though she was exact clone, but it was not very healthy. Or the cloning is now considered a promising tool for preserving endangered species. So now the main utility of this can come for creating for preserving basically any endangered species, like the species which are now on brink of extinction and all. So those species can be now revived or preserved. So this is very important technology for in this particular regard. So basically preserve endangered species and may be important in future production of the transgenic livestock. So now after the success of creating Dolly, somatic cell nuclear transfer was used for creation of several different animal clones. Now sheep was, as Dolly sheep was created in 1996. Then cattle was also cloned in 1998, mice in 1998, goats were also cloned in 1999, pigs were cloned in 2000. So there's so many success rate now. God in 2000, in 2001, cat 2002, rabbit 2002. So mule 2003, deer in 2003. So and now wild animals also, you can see horse 2003. The large animals also were cloned. Dog 2005 was cloned. Parrot, wolf also. Predator also the clone in 2007. Camel also was cloned in 2009. Ibex in 2009. So now you can see so many animals have been now cloned now. So basically this success of this somatic cell nuclear transfer, which was this technology which was used for creation of Dolly the sheep, has been very successful in creating so many animals cloned from different animals now. So now this indicating that this particular technology can be used for saving now endangered species or any livestock or any domestic animal which produces large amount which is highly productive, like produce a large amount of milk, good quality of milk. So those can be now uh, cloned. Similarly, the sheep which provide very good quality of wool, so they can be cloned, so that one farmers can have better return. Pros and cons of cloning: produce animal with desirable traits, as I mentioned, good quality of milk, large amount of milk, good quality of meat, good quality of wool. So those animals now can be cloned, and one can have large stock of those particular uh, cattle with desirable traits. And this will lead to increase in the efficiency of the livestock production, offset losses among endangered species population. Particularly, this is very useful technique, which can be used for preserving or reviving endangered species. Enable better research for finding cures to many diseases. Disease models can be studied here. Providing children for parents who would like a child but can't have one for various reasons also. So this provide parents with an opportunity to clone a child who has died. So these are in future one may attempt these. Decline in but there's some cons. Decline in genetic diversity, taking nature into our own hands. This is religious and moral reasons. Physical problems such as birth defects can occur, just as Dolly, she was um, not that healthy. Possibility of mental emotional problems of the clone. So there are several advantages and disadvantages of animal cloning. The main ethical issues can be against the religious belief. So many communities may not appreciate this effort. So some other advantages can be like people can get an animal that looks like the same as the pet they had earlier. Scientists can clone organs and help people live longer. Scientists can also clone body parts such as ears to surgically attach to a person. Cloned animals are safe to eat also. These are some of the advantages. There are some other risks like but these technology are highly expensive and highly inefficient also. Like success rate is very low. More than 95% of cloning attempts fail to produce a viable offspring. So lot of failure in this technology. In addition to low success rate, cloned animals tend to have more compromised immune function and higher rates of infection, tumor growth, and other disorders. So this problem may be there because the cells are taken from adults, so they are already aging cells. So that's why they may be having a lot of defects or chromosomal abnormalities or immune function defects can be there. Many cloned animals have not lived long enough to generate good data about new clone age. So these are some of the issues which needs to be addressed while fine-tuning the process of animal cloning. But it has a bright future. Scientists have accelerated more into the world of animal cloning. Since Dolly, the first cloned mammals, was created, scientists have cloned many more of the mammalian species such as cats, horse, pigs, cows, and big supply of rodents. But where it is going, for a few hundred years, scientists have tried to protect endangered species now. For this is a very useful technology to protect the endangered species. Because as I mentioned in earlier slides, wolf and others, camels, they have been cloned now. Why not just clone them? Want to bring back the dinosaur? All these are possible things. Okay, so the process of copying an animal could indeed do miracles then. So scientists also can do this also, plants rooting out the disease of the donor animals by using DNA transfer technique. Using these techniques, they also plan to make better meat and food carried by the livestock. So all these are possible now using animal cloning now. So thanks.